And today, as Lionel said, I'm going to talk about the diver. And the aim to build this microscope was to go deeper, image deeper in turbid media. And the developers of this microscope were Sasha, that you can see him there, that he retired. And that's why I'm doing this presentation, but I have been his disciple this past year, so it's an honor for me to show you how amazing this microscope is. And as I said, it was a development with Sasha and Enrico and many other people. So sometimes in biology, we need to image through samples that disperse light, and the light travels differently in samples where there is a scattering medium. And this makes things more complicated. So here I'm showing a real example that sometimes you need to image through like a whole animal. Here is a newborn mouse. It's a real example done in the diver. And in this case, was needed to image the skin of this animal. So you can see how challenging these things can be. So how to do that, how to handle these samples? Well, the LFD developed the diver, that stands for Deep Imaging via Emission Recovery. It's a microscope that is capable to image in turbid media with high resolution and in depth of a few millimeters. So the diver has many capabilities, and we can have, I'm going to put the pointer. We can have an increased depth, we can go deep, we can have a wide field of view, we can do FLIM, harmonic generation imaging, and hyperspectral imaging. And during my presentation, I'm going to talk about all these capabilities. And hopefully, by the end of the talk, I can convince you how powerful and strong the diver is. So the diver is based on a special detection system, but it's based on a two-photon excitation. So when we do two-photon excitation, in order of two photons to be absorbed, we need to have high-intensity light in a very small point in order to these photons to be absorbed. So because of this process, it depends highly on the excitation light, on the to the square of the excitation light. We will only have this excitation in a very small point that is called the point spread function. And this small point is what's going to give us a high resolution when we scan through the image. So using two-photon excitation has some advantages. There is a high penetration of red light in the tissues, and there is less scattering for red light. And also there is less absorption of molecules that are present in the tissue, like hemoglobin, melanin, etc. And in the near infrared area of the spectrum. So this the two-photon excitation is good to image in biological tissue. So when we excite in scatter media, there are two types of photons going to the excitation point. Some photons are going to be unscattered and are called ballistic photons and are going to go to the excitation point straight. And there are other photons that are going to be scattered through the sample, and eventually they have the probability to go to the excitation point, but they will not arrive at the same time in the same space. So those scattered photons are not going to induce two-photon fluorescence. And that's very important, because if all the areas would induce fluorescence, we would broaden the point spread function and we will lose resolution. So the key point is that even in turbid media, we can have high resolution images using two-photon excitation, because only the unscattered photons are going to be able to excite the sample. But there are some limitations on how deep we can image in the sample. And this is because the ballistic photons are going to be attenuated with depth. So the deeper we go, more attenuation will be. So to compensate for that, would increase the excitation light. But there is a limit on how much we can increase this excitation light, because we will start to have high intensity light at the top, at the surface of the sample, and this signal will mask the signal coming from the focal point. And there is other limitations that are, for example, when we have different areas with different index of refraction, that this will distort the optical path of those ballistic photons, and they will not arrive exactly at the same point, and this will broaden the point spread function, giving us a less resolution. So these are some limitations that we have. So the a common configuration for two-photon microscopes are the epi detection, where we have the excitation through the objective lens, and then the fluorescence coming from the sample will go back through the objective. The excitation light will be separated from the fluorescence using a dichroic mirror, and then this will direct the fluorescence to the detector. Here, when we have scattered samples, the photons will come in very different angles, but the objective lenses will only collect fluorescence from a small area from the sample, and many of the photons will be undetected. In the diver detection, we use a wide area detector that is 1 inch square, and this will allow all the scattered photons to reach the detector. And also the other key point of the diver is that we match the index of refraction from the sample to the detector, and the more refractions you have, the light will be attenuated. So in this case, we minimize the losses of photons. And in our case, the photons, as I said, will come in many angles, so this will be an advantage. And here I'm going to show small example with the refractive index matching principle. So here is a real example where there is this printed LSD, and this was imaged through glass slides with air gaps in between, and you can see how the image is not very bright. But if we try to match the index of refraction even with water, that it will have a more similar index of refraction of glass compared to air, we improve the signal coming. So this is the principle where the diver is based. Also, when you have photons coming from many different angles, some of them, because of what is called total internal refraction, many of the photons can be completely refracted and and lose them. So this proves that we could prove that the diver has like a 3 orders of magnitude more signal than the epifluorescence construction, and this has very few optics, and it's a simple construction. So here you can see the diver schematics. As I said, we use a 2-photon laser. We have an acoustic optic modulator to change the intensity light of the excitation beam. We have the scanner, the X and Y scanner, scanning optics, and then I'm going to make a point about how these are in the diver. And then here we have the objective, and here we have the sample with the diver detector. And this is connected to a film box that allows us to do lifetime measurements. And the stage is an XY stage that has a 10 nanometer precision that allows us to do 3D imaging. So if we focus on the detector, here we have a wide area detector. As I said, it's 1 inch square detector that is operates in photon counting mode, this photon multiplier. Then here we have a chamber where we have the filter wheel that are optical filters, because in this case we cannot use interference filters. because the photons come in many different angles, and the interference filters, they need collimated light. So all of these are optical filters. And then there is, in this metal chamber, there is a liquid to match the index of refraction, as I mentioned. Then we have the BG39 filters, that are filters that will allow the visible light to go through the detector, but it will cut the near infrared light coming from the excitation beam. So that light will not reach the detector. So here I'm showing the divers that exist in many different places. Since the diver is the detection part can be put in commercial microscopes, and there are many already built at the at UCI. We have 2 of them. There is one in Colorado, one in Georgetown University, where Suman is, and the good part is that you guys will have very soon one in Uruguay that you'll be able to use there. So going through other of the capabilities, we're going to talk about the deep, how deep we can go. So here the diver has also an epi detection. So this allowed us to compare the diver versus epi. And here there is a phantom, brain phantom, that mimics brain tissue, and there are fluorescent beads inside this phantom. And here is imaging using the diver detector versus the epi, and we compare both of them. Here you can see how after 2 millimeters, we can still see the beads using the diver, but with the epi fluorescence detection, we cannot. And here there is a comparison of a ratio of the epi versus diver signal, and you can see that in transparent medium, even the diver is better because the numerical aperture of the objective lens doesn't change, but for the diver, the effective numerical aperture increases the deeper we go. So it's even better the deeper we go. And for scattered samples, the diver has 650 times more sensitivity than the epi detector, and as you can see here. So here there is a mouse brain tissue, where also we can see here how in epi detection, after 500 microns, we cannot see signal, and with the diver we can. But if you pay attention, here in the deeper part of this image, it's a little blur. And this is because of the changes of index of refraction areas that they deform the wavefront. So they make the image blur because the point spread function broadens, as I mentioned. So one way to overcome this is using adaptive optics. Former PhD student of the LFD, Simon Lemans, worked on this project, and he developed these adaptive optics in the diver, where using a deformable mirror, you can compensate for this wavefront deformation. And here you can see with the AO off, using the diver detector, or when it's on, and you can see how you can gain resolution and go deeper in the tissue. So how wide your field of view is depends on how much you can tilt the beam across the objective aperture. And in the case of the diver, the scanning lenses are very big, and they allowed a wide angle to tilt, and this allows to image very broad fields of view. Here you can see a mouse bone that is made doing multiple tiles, but each tile is 1 millimeter, which is really nice. And this is using a 20x objective, 0.4 numerical aperture. But using a 10x objective that we have, we can go up to 3 millimeter field of view. So this capability of the diver is very nice when you are trying to image big areas in tissues. So moving to the other capability of the diver is the film. So Stuman yesterday showed you this experiment with these mouse livers with non-fat diet or Western diet, and these images were done in the diver. And here we can combine doing film and also having a wide area and being able to image a full liver. Also, as I mentioned, we can do 3D film because of the XYZ stage that we have. And here you can see is an image of a mouse liver. And in the intensity image, we cannot see differences in features, but when we color the spacer plot, we can see different features in this tissue, and the same here in this mouse embryo eye. And not many microscopes can do 3D film, so this is a very nice capability. So other capability of the diver is the harmonic generation imaging. The second harmonic generation, it's a nonlinear optical effect that is generated by specific structures that they lack the center of symmetry. And this effect does not involve absorption, and it doesn't require staining to do this imaging. It's just like the intrinsic properties of these molecules. So when you excite with certain wavelengths, these molecules, the light coming out of it will be a light of double the frequency and half of the wavelength. And it's forward directed, so whatever is the direction of the excitation light, it will be the direction of the light that we collect. And the diver is very good in this sense because of the transmission geometry of the construction. So since of this direction, we will collect all the photons coming from the second harmonic. So that increases the sensitivity versus other microscopes trying to do this second harmonic generation imaging. And here we can see some examples that were done at the diver. Some molecules that can produce this second harmonic generation are collagen or myosin. Here we have a muscle image, and here we have collagen fibers in tissue. Other possibilities to have third harmonic generation, that this is generated by other types of molecules, and it's when you have or structures, that is when you have changes on very sharp changes on the index of refraction, that happens with lipid droplets, in cell membranes, etc. And in this case, when we shine with certain excitation wavelengths, what we would collect is a third of this wavelength. So in the case, if we excite, for example, with 1040 nanometer excitation, then we will collect light of 347 nanometers. And this is UV light. So this will be a problem because you need UV optics, objectives, dichroic mirrors, etc. But the diver, as I showed, is a very simple construction and doesn't have much optics in between the sample and the detection. So for us, it's not a problem. And also, our detector can detect light from 300 nanometers to 700, so we can perfectly collect this light. So here we have some examples that were taken at the diver of adipose tissue. Here we have a liver with non-fat versus fat diet. All this is not stained, it's just the tissue, the properties of the tissue that can give us these images. Here we have a sciatic nerve. Here in red we have the third harmonic, in green second harmonic, and in cyan it's just the autofluorescence of the tissue. And also here there is a bacteria, E. coli 3D image using third harmonic generation. So moving to the last capability of the diver is the hyperspectral imaging that we can do. You heard about hyperspectral imaging in Lionel's talk yesterday. So hyperspectral imaging is when we can have the information of the emission spectrum in every pixel of the image. So in the typical setup of hyperspectral imaging in commercial microscopes is that you need to have a prism gratings to separate the light, and usually you have like a detector array to collect this light. But in these cases, these gratings, they need collimated light. And in our case, that we are imaging like scatter samples, this is not the case. So the LSD has put a lot of effort in trying to do hyperspectral imaging in a different way to be able to use it with light that is not collimated. So I'm going to go fast over the spectral phasor approach because you learned about this yesterday. But in the phasor approach, we can transform the emission spectrum on every pixel of the image in one point in the spectral phasor plot. And in a simple way, the spectral transformation is a mathematical transformation where we multiply the profile of the spectrum by a cosine or a sine function, and we divide by the total intensity for normalization, and with this we can obtain the G and S coordinates of the spectral phasor plot. And each point in this phasor plot, the angle will depend on the center of mass of the spectrum and how close each point is to the center. The modulation will depend on how broad the spectrum is. So in 2018, our group proposed an alternative method to do hyperspectral imaging. And instead of measuring the real spectrum in every pixel of the image, in our case, what we do is to image through filters that have the cosine or sine function profile in the transmission, and we image through these filters. So as you can see here, we have cosine and sine, and what we do is like instead of measuring the real spectrum, we image through these filters, and it's the equivalent of doing that. So we have to also do a measurement of intensity for normalization, and having these three images, we can do calculations and plot the points in the spectral phasor plot. So in this case, we can use scattered media. This is cheaper than spectral detectors, but the point is that we need 3 consecutive measurements in the diver detector because we have only one channel. But this was a very nice development of the LSD, and here I can show you how we have fluorescent beads, and they appear in different parts of the spectral phasor plot, and we can separate them in the images. And here we have a convallaria sample that is a root of a plant, and here we can see different features in this tissue. And also here is a mouse bone with also the same features that have different emission spectra. So the diver, I want you to remember these 3 consecutive measurements for the second part of the talk. So there have been many publications using diver detector in the past years. So there are many applications that the diver can be useful to study. But this is, we were trying to improve the diver. There was a point where we were like, okay, how we can improve this? Because as I mentioned, when, for example, we're trying to do hyperspectral imaging, we need to do consecutive measurements. So we need to do 3 measurements, and if we are trying to use live samples, they move during this time. So what we tried to do was, okay, how can we do it better? So we generated a new detector that is a photomultiplier with 4 channels. And this photomultiplier, it has it can collect signals from 250 nanometers to 700. And this will allow us to do multiple measurements at the same time. And we connected this detector to a film box with 4 channels, so we can do film also in every channel. And we also had to generate the software to collect that and analyze that data. But in this case, having 4 channels will allow us to, in one shot, have multiple information. So we can have the same capabilities as the diver, but in this case, we will have everything at the same time instead of having to do consecutive measurements. And this is very important when you are trying to do metabolic imaging in live samples. And also, even if the samples are not live, it's time consuming, so this also would shorten the imaging time. So all these that I'm going to show now about this new development hasn't been published yet. So I'm showing fresh results of all this. And here we have the same microscope layout as I showed before for the diver, but we have to pay attention here in the detection part. And also we are connecting to the film box 4 independent channels. So if we see here the construction of the new detection system, we have the sample on top of the BG39 filter that, as I mentioned, allows the visible light to go through and reach the detector, but blocks the excitation light. Then here we have a shutter. And then one important thing is that we have to add a diffuser. So all the photons that come out of the sample will be diffused and are able to equally arrive to the 4 different channels of the detector. And this detector is as wide as the diver detector, is 1 inch square. So we have still this wide area detector to collect all the photons. And then we have a filter plate here to separate the fluorescent light, and I'm going to go into detail of that. So as I mentioned before, in this case, we need optical filters because we cannot use interference filters. And when I'm talking about that, here I have, I don't know if you can see, but we use Amazon filters that they sell for photography purposes. So are these thin filters that cut light of different wavelengths, and we put it in front of the different channels. And these filters, they have a profile that resembles the cosine and sine functions, as you can see here. And we have another channel for the total intensity. So with all these, we can do hyperspectral imaging, as I mentioned, and it will be just in one shot. We can calculate the spectral phasor plot. And then we have another channel that we put a filter here, the UG11 filter, that it's good to collect the light coming from the 2nd and 3rd harmonic generation. And we can obtain also corroborate with the lifetime measurements that they will have 0 lifetime as expected for harmonic generation, because as I mentioned, it's not does not involve absorption. And in every channel, we will have the information of film. So the filters that we are using are not ideal with the sine and cosine function. But here we have simulations of different emission spectrum along the visible spectrum. And you can see the positions in the phasor plot for the ideal filters versus our filters. And you can see that the positions are a bit different, but we can apply corrections to relocate the points to the expected positions in the phasor plot. Here we have the ideal filters, our filters, and what we obtain, the spectral phasor plot that we obtain with the correction. So here we go to the real data. We use a monochromatic light, different bands from 420 nanometers to 590. And here you can see how the light appears in different positions of the phasor plot and are like all distributed perfectly well. So we can separate all this light of different wavelengths. And here we were testing the hyperspectral imaging part. So here we use different dyes that have different emission spectrum. And we image using our multi-channel, and we see they appear in different positions of the phasor plot as expected. And we compare our results with the results obtained using the size LSM-880 with the spectral detector. And we can see that we obtain similar results as this established technique that is the LSM-880. So then we wanted to test if the linear combination was maintained. That is very important. You already heard about this linear combination property of the phasor plot, that if you have different emissions, different species emitting with different spectrum, they will appear in different positions of the spectrum of the spectral phasor plot. Like here we have coumarin 1 and rhodamine 110, and then we did different ratios of these two dyes, so they are mixed in the pixel. So these pixels, or they have the mixture, will appear in a line that in a line combining the individual positions of the dyes. And in this case, we see that what happens here, we have the points with the correction that we apply, and here we show the raw data without the correction. So we see that the correction is not messing up the linear combination properties of the phasor plot. We did in the same experiment, we checked the linear combination of the film, and we can see that that's also maintained too. Then we wanted to move to a biological system to test our multi-channel detector. And you have seen yesterday with Michelle Digmans and Suman Ranjit talks about this NADH film metabolic imaging, where we have this NADH free and bound NADH with different lifetimes, and they generate this metabolic trajectory that can give us information about what's happening in the metabolism of cells and tissue. And for all these experiments, what people usually do is to use a bandpass filter that collects the fluorescence from 400 to 500 nanometers. So you cut other fluorescence autofluorescence signal, for example, coming from FID. But in our case, we have only 4 channels. We already have occupied these 4 channels. We didn't have another channel where we can put a specific filter to collect NADH signal to do these metabolic studies. But our cosine filter has a profile that collects signal in the same range of the spectrum, but is a bit broader than what is usually used to collect NADH signals. But we wanted to prove that using the cosine filter and measuring film in that channel, if that would be good enough to do this NADH metabolic imaging. So what we did to prove that is we use live cells in culture. We have a controlled situation, and we see the phasor cloud in the metabolic trajectory. And then we treated the cells with different drugs, potassium cyanide that blocks the electron transport chain, or 2-deoxyglucose that also blocks the glycolysis metabolism. And in this case, we block the OXPHOS, and we see that here the phasor cloud moves to glycolysis. And in this case, when we're blocking glycolysis, the phasor cloud is moving towards OXPHOS. So we proved that we can see with our system what is expected, and using the cosine channel, it would be good to do an ADH metabolic imaging and do this type of studies that you have seen yesterday. Another thing that we did was to mix FLIM, once we proved that we can do the autofluorescence FLIM, with second harmonic generation. So what we did is we built 3D cultures with collagen, cells in collagen, live cells. And as you can see here in the images of FLIM or second harmonic, you see there is no bleeding through of the second harmonic on the FLIM channel. One thing that I forgot to mention is that we put a UV absorber coating in our filters to cut any possible signal from the second harmonic in the other channel. And you see that that worked very well for us. And here you can see in the phasor plot that the cells, autofluorescence of the cells appears in this metabolic trajectory, and the second harmonic appears with 0 lifetime as expected. Then we went to mix FLIM and hyperspectral imaging. So here we have a first example where we stain 3 organelles of the cell. We have the nucleus, the mitochondria, and the cytoskeleton, and we have the spectral phasor plot and the lifetime phasor plot. And using our Gaussian mixture model method that we published with Alex and Enrico last year, that is an automatic method to identify phasor clusters in the phasor plot. We can identify that in the spectral image or in the FLIM image, the same features, these 3 organelles. So we are obtaining the same information using hyperspectral of FLIM, using our multi-channel. And here we have another example about Lordan. I don't know if you heard about Lordan yet, but you probably will along the course. And Lordan changes the emission spectrum depending on the environment. And we know that Lordan emits more blue when it's in the membranes. And here we can see with the phasor plot or the FLIM that we can see those spectral changes or lifetime changes in these images too. So we could corroborate that our FLIM and hyperspectral data that we were obtaining was correct. So I don't have much time, but I will try to go quickly to the last example where we try to combine all of the measurements, the second harmonic, FLIM, and hyperspectral at the same time. And this was in a collaboration that we are doing with the Hospital de Clínicas in Montevideo, with Leonel Malacrida and Julio Magliano, where they provided us samples, skin samples, where they have basal cell carcinoma. And these samples are very good in the sense that they already did the hematoxylin eosin staining, and we know where the tumor is located. So we have unstained samples that we can image with our method and try to compare the results. And we're trying to identify in these samples with our method, we can identify the areas of the tumor. So these samples are fixed, and that has some disadvantages in some sense because all the metabolic measurements are done in fresh tissue, but there has been some publications saying that the metabolic signatures are remain in the tissues when after fixation, And also, like some important point is that in tissues which in the area of NADH emission spectrum, there are more molecules contributing to the fluorescence, like collagen, elastin. So it's not so simple how to interpret the information, because there is more molecules contributing to the autofluorescence. But quickly, I can show you here how we image through a sample, unstained sample of these tumors, and here we have the hyperspectral imaging, FLIM, and second harmonic. And you can see when we combine all the information with these three together, we can see the areas that we can limit this tumor. This is preliminary data, so we're still working on this and trying to combine all this information. So one last thing is that the filters that we picked are arbitrary, and it's what we need in our case, but there are other combinations of filters that you can use, and we publish another method of doing hyperspectral imaging using these linear filters instead of the sine-cosine filters, and that can be another option to use less channels and be able to still do hyperspectral imaging.